Hello everyone, and welcome to Reduce Medical Image Management Pain Points, Medical Devices, presented by Medidata. I'm Carly Helfand, Senior Editor at Fierce Pharma, and I'll be moderating this webinar. Our speakers today are Troy Schneider, Principal Medical Imaging, Medidata Solutions, and Nick Wharton, Senior Vice President, Medical Metrics, Inc. You can read their full bios on the left side of your window. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the Files button at the lower left of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We'll follow the presentations by Q&A session. Please submit your questions during or after the presentations, clicking the Q&A button in the lower left corner of the screen. Okay, now let's begin. Troy, please go ahead.
Thank you so much, Carly, for that introduction and for putting this forum together. Welcome, everyone, and thank you for joining our overview today on the use of technology in reducing image management pain points in clinical studies. First, let me say good morning to everyone. I hope you all had a wonderful summer. I really want to thank you for joining our session today. We're going to cover a lot on image management, and we'll be sure to provide some time at the end for questions, so please submit your questions if you get a moment. We'll also provide our contact information at the end for any follow-up discussions, or if you'd like to dive deeper on any of these topics with myself or with Nick Wharton of Medical Metrics. But first, let's do some introductions. I'll go ahead and introduce myself. I'm Troy Schneider. I was the Global Director of Business Development at Intellimage, who you may or may not know of. We were a leader of image management software for clinical trials, and we also developed some pretty slick technologies for pre-surgical planning that allowed device reps in the field to collaborate on complex cases with clinical specialists back at the corporate office, referring physicians, and surgical teams, again, ahead of procedures. Our technologies, our company Intellimage, were acquired by Medidata Solutions in April of 2016. I've been lucky enough through both our portals to have unique experience working closely with sites, sponsors, CROs, and both academic and commercial core labs, really focusing on improving imaging workflows. So that being said, I'd also like to introduce a great friend and a thought leader in this space, Nick Wharton.
Hello, everyone. Good morning. I've really been looking forward to speaking with you all today. As Troy mentioned, my name is Nick Wharton. I am a co-founder and Senior Vice President of Business Development and Strategic Partnerships at Medical Metrics. We're a medical imaging core lab located in Houston, Texas, near the Texas Medical Center. We specialize in medical device trials, and we've been providing imaging core lab services for over 15 years now across a range of therapeutic areas.
Thanks, Nick. So today we're going to focus on covering the following topics during this session. First, we're going to look at current pain points in the industry. Then, how can current or upcoming technologies resolve or improve these pains? We'll then get the perspective of the core lab's process and what they see as pain points through Nick. However, I thought it'd be helpful for us to start by reviewing just a little bit about Medidata. Some of you in the device world may or may not be familiar with who Medidata is. So before we jump right in, I want to take a moment to introduce our company. Medidata is a leading technology company specifically focused on cloud software. We are solely dedicated to helping transform clinical research for sponsors, CROs, sites, and core labs, but most importantly, just like everyone on the phone, the patient. This is what we do, and it's all we do. In fact, Medidata's technologies are involved in over 50% of all electronic-based clinical trials, many of these being medical device trials. The overall goal that Medidata drives towards is to create value by really focusing on three areas. First, reducing cost. Second, lowering risk. And third, let's really speed up the time for these game-changing drugs and devices to market. One of the ways Medidata is able to drive these goals is its continued investment in game-changing technology. Medidata has a commitment of over $120 million on an annual run rate in technology investments. This includes research and development, bringing in new tech talent, and always looking to partner and make acquisitions like the Intellimage one earlier this year. These investments ultimately improve the Medidata clinical cloud experience for sponsors who touch a clinical study. So what's interesting is Medidata now has software and tools in place to help with the complete life cycle of a study from A to Z. We're only going to focus on the imaging piece today. But I wanted everyone to learn just a little bit about Medidata, its history with IntelliMage, and for you to become more comfortable with Medidata's reach and understanding of the imaging ecosystem. With that being said, let's jump into where we are today with image management. Many people don't realize this, but I'm sure a lot of you on the phone do. Managing medical images and imaging data for clinical trials is not easy. It's far from it. The variety of sites and files and proprietary files and settings and firewalls, protocols, users, imaging charters makes the process extremely difficult and extremely costly. In fact, I'm not sure if many of you knew this, but imaging is the fastest growing data collection point now in clinical trials. In fact, Medidata just did a full analysis and found that over 55% of all trials now have imaging as a primary or secondary endpoint. That number skyrockets for device studies. It's a huge and vital part of most trials, and if not done correctly and smoothly, can bring a trial to a screeching halt or cause major delays or inflated costs. And because of that, our imaging team at IntelliMage, now Medidata Imaging, has spent a lot of time capturing data and feedback from the over 5,000 sites that use the IntelliMage platform on hundreds of projects. In fact, the IntelliMage system in the last 12 months has moved over 400 million images. And when the dust settles on all this data, discussions with sites and labs and sponsors, we are really left with 5 common pain points that touch nearly every study involving imaging. So for those on the phone, I'm sure 1 or 2 of these hit home, or perhaps all 5 you've come across as issues in the study. So the first, we're going to look at study delays, then a disconnected process of having to use multiple systems, the lack of visibility. So if you're a sponsor or a CRO or a lab, it's really difficult at any time to easily click a few buttons and see an entire report on where all your imaging visits are across the entire process at any time. 4, de-identification issues and concerns, and 5, study costs that are driven by the imaging needs. So let's start with a review. I think it'd be helpful to start with the current process in today's trials and the challenges we all face. So this graphic illustrates really the process for manually shipping data. But it also could just apply to using what I like to call old school methods of just making an FTP connection between a site and a lab and just simply digitizing this process. So a majority of these pain points when we walk through this exist if you're manually shipping or just use kind of a clunky one-to-one connection. So for those that aren't familiar with this process, I think it's helpful to see where we are today. So first, a subject comes in for either a screening, procedure, or a follow-up visit where they have imaging taken. The study coordinator at the site gets notification that that imaging's done. They actually have to place an order or pick up a phone to call radiology and say, hey, I need the scan for Troy Schneider for a research project. Then the radiology group has to go in their PAC system, find the patient, then they have to burn a CD oftentimes. It then gets couriered up a few hours later back to the study coordinator. Study coordinator then gets a form out, has to fill out a form, has to label the CD, and then has to give it to the shipping team. Then and only then is it put in a pile that should be shipped off to the core lab. Core lab then opens up, makes sure there's no obvious errors, does a quick spot check, then it goes to administrative quality check, and then finally is loaded in for the reviewer. So it's a pretty convoluted process that takes a lot of time and there's a lot of people involved. I'm going to let Nick add a little bit of commentary on the core lab's perspective on this. Nick?
Yeah, thank you, Troy. Ideally, as the core lab, we want to see images with clean labeling, no missing or non-compliant images, and all images being nicely organized and labeled on disk. Now, that would be a perfect world, but this is difficult to achieve with traditional methods. Overall, the biggest issue, in my opinion, is that there's no structured interface that guides sites through the image transfer process. Commonly, when shipping images to the core lab, we need the sites to complete an image transmittal form that identifies the contents of the shipment. Unfortunately, when completing the form, it's easy for the sites to make mistakes, such as transcription errors, or even send us the wrong images. And this puts the burden on the core lab, medical metrics, to identify these mistakes and then resolve them with the input from the site, the sponsor, and or the CRO. So we all have to work together to resolve the discrepancies, and this can be time-consuming.
That's a good point, Nick. I think this is even more magnified in device studies where one study can have multiple core labs involved. Traditionally with pharmaceutical studies, you see one core lab doing a majority of the reads, even multimodality reads. With device studies, you could be sending your echoes to core lab A, your angios to core lab B, and your CTs to a university center. So that's even more compounded using all those different systems with all the different core labs on the receiving end. So after the dust settles, when you look at that spaghetti bowl of a process, we're left with 5 sticky pain points that sites, sponsors, and labs share with us on a daily basis. These can completely disrupt a study, and this is something, honestly, that the industry needs to evolve from. Keep in mind these issues apply to both manual shipments and to the conventional electronic systems. First, manual submissions are killing the speed and power of automated real-time image transfers. Second, we're going to review manually entering data to forms, especially when the data already exists in another system, is the biggest waste of time at core labs and the #1 driver of imaging queries. Third, no or incorrect de-identification occurring, and this is becoming more and more of an issue. Fourth, sites are still sending images that do not match the protocol or aren't meeting the core lab standards or imaging charter. And finally, lack of integration. With all the systems in use, wouldn't it be nice if all the clinical trial systems talked and worked together? This would make the process so much easier for the site, sponsors, and ultimately the patients. So now let's dive in and see what's really going on with these issues. So pain point 1, let's take a look at our first culprit, CDs. CDs were an amazing technology in 1996 when you wanted to go to the mall and pick up a new album, or were you excited to get a CD of 1,000 free hours on AOL dial-up sent to you in the mail. But 20 years later, to use CDs to move clinical data quickly, efficiently, and compliantly, no thank you. And many of these pain points also apply to the clunky one-on-one FTP connections. They're slow, they break, they get scratched, they're sent to the wrong person, and they can't provide any real-time communication. I think for some device examples, let's say you need to quickly send a CT scan for maybe a AAA study to 5 independent reviewers for a screening visit. Can you do that with 5 different FTP connections or with mailing CDs? Maybe you need to send an echo for a serious adverse event to a clinical events committee for review within a certain time frame. Not possible. Same if you have 2 radiologists reviewing an angio and you need to queue up a 3rd for adjudication because of a discrepancy in a timely manner. It's just not possible with CDs. The CDs are holding this industry back. Pain point 2 is data entry errors. As fun and as exhilarating it is for us and sites to enter forms all day, it's a recipe for mistakes. We've all seen some systems that ask sites to fill out your traditional image transmittal forms or data transmittal forms. Essentially, these systems are asking site coordinators, who if you've ever been to their office, you can tell they have 1,000 different things going on at any time. You're asking them to correctly fill out by hand a trial ID number, their name, subject ID, what time point it is, what requirement this is. You're really putting them in an unfair position asking them to do this every time. Alternatively, though, having these forms auto-completed with data points we already know is much more powerful. Even more so if you can leverage the data already exist in the DICOM headers. Creating scripts to auto-complete these common imaging forms has actually led to a 600% reduction in queries, a big milestone. So Nick, I know you guys deal a lot with data entry errors and versus some of the traditional methods. Why don't you chime in on this?
Yeah, thanks, Troy. I think a big gap is not being able to parse the DICOM headers for information that can help detect potential data entry errors. Most people are not imaging experts and don't necessarily know if they are selecting all the appropriate images from the PACS system to send to the core lab. Automated inspections of the DICOM headers could make it easier to identify missing images or even prevent images from being assigned to the wrong subject. Similarly, not leveraging the DICOM headers makes it harder to identify errors in the visit date. For example, the visit date entered by the site may be very different from the image exam date in the DICOM header. Ideally, this should flag an error. Also, without interrogating the DICOM header, it is harder to catch visits that are chronologically out of sequence or out of window. So missing images and incorrect visit dates are some of the most common errors that we see that could be addressed through inspections, automated inspections of the DICOM headers.
You're absolutely right, and that's something I don't think a lot of sponsors know the advantages of doing that, so thanks for adding that. I think without this automation in place, it's easy to see the errors that can occur and how a 24-month trial, or for cardiovascular trial, a 60-month trial, can easily turn into a 72-month trial, and that's not in anyone's best interest when that happens. So issue 3, and probably the question most commonly asked by sponsors, how can we handle de-identification? The current model for most sites in larger hospital systems is not something our industry should be proud of. Most site users, when getting ready to load an image for a clinical study, are required to send it to a separate group somewhere on the hospital campus. So it's couriered over. This group just focuses on de-identification. So your CD with your images and some pretty key data goes into a big pile at a de-identification group's queue. Once they get to it, they then open it up, wipe out the DICOM headers, look for any reports that should be in there, do any masking. They then put it back in a big pile to be sent back to the study coordinator to transfer to the lab. This could add hours, if not days, to the process if they're backed up. And that's only for sites for your really big institutions. For sites that don't have this process, they do the best they can, but they're basically crossing their fingers and hoping for the best. And they send the images off, hoping they're clean, but removing all PHI is a difficult task. The query rate skyrockets to account for these images getting to groups like Medical Metrics or other labs with PHI. This is a big issue. Pain point 4 is no edit checks to ensure imaging data is complete and correct and ready for the reviewers. It's interesting that EDC systems do a great job of providing edit checks on data going into the database. An EDC system can make sure the data is correct, fits the protocol, meets all the parameters before being stored. So why can't we do the same with imaging? With no image data edit checks in place, we see images that don't match the protocol. Maybe they're the wrong subject, the wrong visit, maybe the slice thickness or the MR sequences are off, maybe the echo clip length is too short. We just don't have a good way to catch these, and thus they're getting to the radiologist and they're not ready to be reviewed. And the radiologist piece is one of the more expensive parts of a study. So we want to make sure and try to really work on using technology to improve cleaning these images before they get to their final review, because currently this is adding a tremendous amount of time and money to the process. Pain point 5, and one that's mentioned mostly by sites and even more so lately by core labs and CROs. The number of different and siloed systems they have to use to just move the imaging. It kind of goes back to the graphic we looked at earlier. I mean, keep in mind, the study coordinator, when they're getting ready to send an imaging visit, has to call radiology. The radiologist team has to look in their PAC system. They have to pull that data out, burn a CD on another piece of software. Then it goes to that de-identification team who has to use a de-identification program to de-identify. Then it's sent back to the study coordinator to use the image transfer tool or to ship it. Then the lab has to do an administrative quality check in a system. Then they put it in the viewing tool, and then the data is entered into an EDC. So it's a little dizzying to catch up with, but let's just say we just moved one x-ray and we had to use 6 to 8 different systems. That burns a ton of energy, causes a lot of frustration with the sites having to remember username and passwords for all of this information. You go into these sites and they say, oh, that's our computer for that system. If you go down the hall, take a left, go up the elevator, that's our computer for system 2. This is just causing so many frustrations and delays. Now think if we could all accomplish if the clinical trial systems were all connected and integrated. So after looking at these 5 pain points, I'm sure some of them have hit home. These issues have actually led the industry to an average of just above a third query rate on images, so 35%. Having just over a third of all imaging submissions have an issue is a big issue. But let's flip the script a little bit. Let's relook at these issues when we have technology as an ally and have cutting-edge systems at work. Let's see what we can do with that 35% query rate when you have a tool like the Medidata imaging solution driving the image management. So for those on the line that weren't familiar with IntelliMage, despite a very big device footprint, after the acquisition, Medidata Imaging has created a wonderful system, a cloud-based image management system that is really powerful at moving images from the sites to the labs, but that's not what we focus on. We've really focused our time, our energy, and our technical resources on creating a system focused on integration, focused on powerful de-identification, edit checks to make sure perfect images are getting to the reviewers, and workflows to handle any of your trial designs with ease on a configurable platform. But we can still move images quickly, reliably, and they can be in DICOM or non-DICOM format. Just allow sites around the globe to easily, without install anything, fire up any browser and send de-identified images to the reviewers quicker than ever before. They're then automated at download at the core lab or into the viewing tool of choice. So we've worked hard to build this system and remove some of the pain points we've talked about the last few years. So let's take a look at how we've done that. First, let's have a system that focuses on removing manual entry so we can reduce all those data errors. So instead of asking the site user to type in or handwrite the trial ID, their name, subject ID, visit requirement, let's actually have them select the study, log in, select the study, select their subject, and then easily select their visit in the bottom right, and then the requirement they want to upload. Then they can easily upload in the background, and we'll actually log all the data in the audit log and pull out the DICOM headers and auto-populate forms with it. So it's easy to navigate, very little room for mistakes, easy to use, and takes 10 seconds for manually completing your image transmittal forms. Then we can start to really do some cool things like auto-completion of important forms with data from the audit log and data from the DICOM headers. These forms can have real-time edit checks and even change the routing of the images based on the answer. So imagine a scenario where if intravascular thrombus or a question was answered on a form, maybe you want a different key opinion leader or somebody on your clinical events team to receive an image right away when that's noted on a form. That's some real power, and don't forget the integration piece. Let's have these forms show up in a report, and let's send this information back to an EDC system like Medidata's Rave. Then after uploading the image, let's remove mistakes from the data entry and use advanced technologies to help with de-identification. The Medidata imaging technology allows for custom de-identification schemes per study. And the beauty of this is it's set up in advance, so when users don't have to worry about it. When they click upload, the Medidata imaging technology takes over and does all the de-identification. We even have technology to identify and remove structured reports, radiology reports, overlays, and PHI that may be burned into the image. Once these images are de-identified and match protocol, we integrate with the core labs like Medical Metrics to seamlessly download the images right away in near real time, right into the servers or tools they like to use. This is one of the biggest savers in time. This way Nick and his team aren't diving through different folder structures looking for images, or different images are going to different computers. They all seamlessly integrate into the tools they like to use. Also focusing on integration, we have the ability through our APIs to launch third-party viewing tools that your teams may want to do for 3D renderings or other advanced measurements. We can even auto-detect measurements in our own PACS viewer or in other viewers and auto-populate CRFs within the IntelliMage system or within EDC systems like Medidata's Rave. A really powerful tool to capture measurement data and limit the number of systems. This used to take 4 systems. It can actually be done now within 1 within Medidata imaging. So if we relook at the 5 pain points from earlier, we've taken a lot of issues and now we've, again, flipped the script, and these are nice milestones in the industry to highlight. We've taken trial delays and inflated costs and turned it into improved time to market and reduced costs. We've taken a disconnected process with no visibility for sponsors and connected all the systems so a sponsor can log on and get complete visibility on their imaging at any time. And because of all this, We've actually seen, using the Medidata image platform, the query rate on images drop from 35% to less than 7%. And this is an area we continue to focus on improving on. So that being said, I want to pass this over to my co-presenter, Nick, who's going to share some of the perspective of a core lab focus on medical device studies. So Nick, I'm going to pass you control. And please go ahead. Thank you.
Well, thank you very much for that, Troy. Again, I am delighted to be able to speak with everyone this morning. As mentioned earlier, my name is Nick Wharton. I am a co-founder and principal of Medical Metrics. I've been working closely with Troy and his team for the last couple of years, and I'm pleased to comment on our experience adopting Medidata into our workflow. The goals of my short presentation today are to, 1, discuss MMI's selection of Medidata as our preferred image management vendor for clinical trials and product surveillance. 2, to describe how we use Medidata for image de-identification and handling. And 3, to describe the positive performance gains we have achieved with the system. So who is Medical Metrics? We are a medical imaging core lab located in Houston, Texas. We conduct independent evaluations of product safety and performance from medical images. We support studies ranging from preclinical product evaluations to early phase clinical testing to pivotal studies and post-approval surveillance. I think a key differentiator for MMI is that we are FDA registered and we are regularly inspected in connection with our services and technology. We are also ISO 9001 certified. And it's our commitment to continuous improvement that fundamentally drove our interest in the Medidata platform. We specialize in Class 3 medical devices and biopharmaceuticals. Over the last 16 years, we have supported nearly 500 large-scale clinical trials and research studies in the areas of orthopedics, spine, neurology, cardiology, and vascular medicine. To date, we've evaluated well over 160 unique treatments across these therapeutic areas, and some examples are shown in the graphic on the right. We have a positive product approval history. To date, 20 products have been reviewed or approved by FDA with our support. And within the last 36 to 48 months, at least 7 devices have obtained PMA approval. These statistics reflect that we understand the requirements for high-quality imaging and regulatory compliance, and our use of the Medidata platform underscores this commitment to quality. I would also comment that when preparing to evaluate new treatments, we deliver a high standard of scientific rigor to the study design and planning process. We work with leading authorities in medical imaging and clinical practice to design effective protocols for assessing product performance. Our experts include leaders of professional societies, editors of high-impact journals, department chairs of radiology, and expert advisors to FDA. These experts can only do their jobs, however, if we receive high-quality images from the sites, and this is another motivation for the Medidata platform. We use the platform to support 2 broad categories of services at MMI. One category is our clinical trial business. As a core lab, we use the platform to receive and process medical images from large-scale clinical research studies and analyze them according to a pre-approved protocol. We also use the platform to provide product surveillance support as a secondary category of services. Some of our larger clients with approved products that are already on the market ask us to evaluate images of possible product failures or to investigate surgeon complaints. We use the Medidata platform to receive the images and then conduct specialized evaluations of those images. The scope of the analysis tends to be unique in each case. So let's talk a little bit about how Medidata fits into our workflow. In general, there are 6 basic steps in the image handling process. Image acquisition at the site, image transfer from the site to the core lab, image receipt by the core lab, image inspection and site feedback, image analysis in QC, and reporting of the analysis results. We use Medidata for the image transfer step of the process. We rely on the platform to assure that all images are sent to us electronically in a secure and compliant fashion with minimum burden to the site. The platform has other functionality beyond image transfer, but transfer is currently how we use the technology in our clinical trial business. Historically, before we adopted Medidata, we relied on two basic forms of image delivery: courier and basic electronic transfer. Courier shipping includes FedEx, DHL, and UPS. Basic electronic transfer methods include secure FTP, which is an internet-based file transfer method, and DICOM transfer, which is a method for transmitting electronic images between DICOM compliant systems. Before we adopted Medidata, secure FTP was the most common electronic transfer method that we used. Both methods of image transfer, courier and FTP, require sending a transmittal form with the images to identify the contents of the shipment. This form provides the necessary information to uniquely identify the images for storage in the study database. However, as Troy mentioned earlier, these forms can result in many discrepancies. This is an example of a transmittal form for a hypothetical study at MMI and a sample CD label. The data on the form includes information about the site, the date of shipment, the site number, the name and contact information for the person sending the shipment, the subject ID, the visit designation, the visit date, the treatment group, the treatment location, and other information. It's a very manual process for entering all of this information, and data entry errors are common. Additionally, images that may be placed on the CD or shipped via FTP are not always consistent with the information entered on the forms and labels. And this is because the images and forms are not electronically linked. There are multiple challenges associated with traditional image transfer methods for core labs. Troy has already done a great job describing them, so I won't spend much time here, but in brief, the key challenges are limited or no image de-identification. Without additional software, there's no way to mask or anonymize the images before putting them on a CD or transferring them via FTP. No site-side error detection. Errors in labeling and protocol compliance are usually identified only after the images are received at the core lab. There is no real-time warning to the sites about any potential errors or discrepancies with the data. Paper transmittal forms. As you have seen, completing transmittal forms is a manual process that can be burdensome and error-prone. Multiple steps to receive, process, and store images. When images are received on CD or via FTP at the core lab, there are multiple steps associated with moving the images from the input media into the core lab's database, and then combining the transmittal information with the images. This takes time and must be done meticulously by the core lab to avoid errors. And finally, no real-time access to data. While the images are en route to the core lab, there is no opportunity for third parties, including the sponsor or the CRO, to view the images or see what the sites are sending in real time. The images become visible only after they have been cleared of discrepancies and loaded into the core lab system. From our experience, sponsors are demanding greater transparency and visibility to their data, and our partners need a way to monitor site performance. Given these challenges and pain points, MMI looked for a new technology solution that would address the gaps and pain points in the image transfer process. Our requirements for selecting new technology were these: 100% de-identification of protected health information. We needed to make sure, particularly for countries with strict privacy provisions, that no subject identifying information was included with the imaging. We also needed a way to customize the platform to anonymize the DICOM tags that we specifically chose. And we also needed a way to mask or black out information that was burned into any of the images. Automated inspection of DICOM headers. We needed a way to automatically identify disagreements in the information entered by the site versus information available in a DICOM header. We wanted to work with a group that would be willing to develop custom functionality that could be integrated into the software and allow us to implement very specialized inspections related to protocol compliance. For instance, for studies with advanced MRI, we wanted to be able to implement unique inspections of the MRI sequence parameters. 24/7 site technical support. We wanted a team of professionals available to address technical queries that our project managers and other operational staff might not be able to handle themselves, particularly outside of business hours for sites in distant time zones. Structured electronic data entry. Given the limitations of transmittal forms, we needed an intuitive software interface to guide clinical sites through the upload process and, wherever possible, automatically flag potential errors or warnings before the images were submitted to MMI. Ability to handle large data files. As studies get more complex and as new imaging technology continues to evolve, we're dealing with increasingly large data sets. We needed confidence that the technology could support large uploads. We also needed features for auto recovery in the event of a network interruption, as well as the ability for site staff to walk away from their computers after launching the upload process. Support for in-house setup and configuration. The ability to configure and customize the software was also a critical requirement for us. We didn't want to be dependent on a third party to set up the system and verify the configuration. For studies with rapid startup timelines or complex customization requirements, we wanted to be able to build the configuration ourselves and perform the necessary user acceptance testing in-house to assure that we were meeting the stringent requirements of each trial. And lastly, client and CRO reporting and viewing functions. We wanted to provide greater transparency to our clients and our CRO partners by giving them tools to view the images and generate real-time image reports. Although we routinely provide inventory reports to our clients at specific intervals, we wanted to provide direct access to the data so that users could export a summary report on demand if needed. Troy, is there anything you'd like to add to this?
No, I think that last point is becoming more and more popular in device trials, especially as CROs sort of market share grows in their involvement in device studies and the services they provide. You know, the ability for CROs or consultants to also access imaging reports is really vital. So the ability to run reports out of even your EDC system like Rave and see all your imaging queries, where every image is in the process, where each image is with each reviewer, how long the core lab's taking on each one, I think is a huge advantage. It seems like the turnaround time for reads is becoming more and more quick, and so having access to reports that reflect and help drive that process is crucial.
And thanks for that, Troy. So then, based on these requirements and considerations and all the value added through capabilities of Medidata, we selected Medidata as our preferred solution for electronic image transfers. We validated the technology in late 2013. We made Medidata a qualified vendor and partner in early 2014, and then we rolled it out for use in earnest right after that. This is a screen capture of the Medidata medical imaging interface, which Troy has already discussed. I won't spend much time here except to briefly comment that the interface is designed to minimize the amount of manual data entry and manual error checking required by site personnel. For comparison with the image transmittal form I showed earlier, the red arrows illustrate where the site enters information, selects from a list of available options, and receives feedback on potential errors. The data entry interface is much more streamlined and provides automated notifications that really are not possible with transmittal forms. there is also a mechanism for the site to view images, to upload and verify that the images are correct before submitting them. Sponsors and CROs can view the images in the same way. Now let's take a look at platform adoption rates over the last 2 years from September 2014 to today. Over the last couple of years, there has been strong adoption of the system by the vast majority of our sponsors. Internal data shows an adoption rate of 96% for sponsors of new studies at Medical Metrics. Clinical trial sites have also shown a positive adoption rate of about 90%. When qualifying and training sites on the image transfer process, we have found most sites have been willing and able to use Medidata in lieu of Courier or FTP. To assure adoption, we have actively promoted the platform to the sites, and many, if not most, sponsors have actively encouraged sites to use the system wherever possible. In some cases, sponsors have actually mandated the use of Medidata for image transfers and have not permitted sites to use courier shipping. So working collaboratively with the sponsor, with the CRO and others, we can really promote adoption and really direct the sites to use the platform, which will result in efficiencies across the board for all study participants. Now let's review the effect of Medidata on discrepancies. Over the last 2 years, we have seen a 1/3 reduction in the number of discrepancies for images received via Medidata versus images received via FTP or Courier, otherwise non-Medidata. For images received via Courier or FTP, nearly a quarter had at least one discrepancy related to labeling, missing images, or protocol compliance. We worked really hard to train the sites on the requirements for image transfer, but even in spite of solid training, nevertheless, there was still nearly a quarter of instances where there was a discrepancy in the image transfer. For images received via Medidata, only 16% had a discrepancy. This represents a 33% reduction in the number of visits with discrepancies comparing the two methods. Said another way, 8% fewer time points have discrepancies if they are received via Medidata. And over time, we expect that rate to increase to 10% or more as we implement more features of the system. Briefly, I wanted to review some of the key areas where we have seen reductions in discrepancies. This table shows 4 prototypical types of discrepancies that we encounter in clinical trials. The blue and orange bars show rates of discrepancies for Courier and FTP respectively. The gray bar is for Medidata. One type of discrepancy shown on the far left relates to missing images. Missing images may be related to a missing view, such as a missing X-ray view, flexion or extension, a missing imaging modality, such as a missing MRI or CT, echo, or a missing series or sequence, such as a missing sagittal or axial T2. Finally, and very commonly, images may be missing from the shipment because they were simply never taken. For Courier and FTP shipping, 10 to 12% of discrepancies are due to missing images. And this has been reduced to 7% for Medidata. Some of this 10% is because the images were never taken by the site, so we shouldn't necessarily expect the rate to fall to 0 with the use of Medidata. There's always going to be some number of missing images because those images were never taken. With respect to extra or off-protocol imaging, a 2% discrepancy rate occurs for Courier and FTP, and this rate drops to 0% with Medidata. This is because the system can be configured to prevent sites from sending us non-study images. Similarly, with respect to visit labeling errors, the discrepancy rate drops to 0% for Medidata, whereas the rate is 3% for Courier and 2% for FTP. An example of a visit label discrepancy is where the visit ID, such as pre-op or 6 months, does not match the visit date. Another example is where the visit label on the transmittal form contradicts the label on the CD. These types of discrepancies can be eliminated because the Medidata software can automatically detect them and flag a warning, so the issue can be addressed before the images are submitted. Finally, another type of error that occurs with courier and FTP shipping is a missing or incomplete transmittal form. This type of error does not happen with Medidata because transmittal forms are not required. Reducing the number of discrepancies has streamlined our project management and QC activities. For every discrepancy we can eliminate, we estimate that we can save 25 to 30 minutes on average in resolving that discrepancy. This represents the amount of time needed to document the issue in our discrepancy management system, investigate the issue, report it back to the site as a query, track its status, including repeated follow-up with the site if we don't get a response, which is common, resolve it, and documenting the resolution for auditing purposes. So there's quite a bit of time that's required to resolve each and every discrepancy that we see. Now, we receive and process anywhere between 1,300 and 1,600 time points or more per month, so it's high volume here. And based on current metrics, if we reduce our discrepancies by 8 to 10%, this saves at least 45 hours per month in discrepancy management time. This time savings can help to reduce image lock periods while we prepare deliverables. The time savings can also generate greater confidence in on-time delivery because we spend less time chasing down and resolving queries. Finally, the reduced time resolving discrepancies means our project management team can spend more time on other quality critical initiatives. In addition to the time savings we gain from the reduced discrepancies, we also gain improvements in productivity. For example, MMI tracks how long it takes to import images into our database after the images have been received. Data on image import times indicate that images received via Medidata are imported into our system 50% faster than images received electronically via FTP, and more than twice as fast as images received via courier. Some of the reasons for the faster import times with Medidata are that the images arrive in a clear and well-organized format with fewer discrepancies. This makes it easier for the team to select and load the images. In comparison, if images are received on CD, handling the physical media and the printed transmittal forms is less efficient and difficult to automate. We've tried to improve the process by putting barcodes and QR codes in our transmittal forms and labels, but the process is still much less efficient. Another reason we can import images faster when received via Medidata is because we use a software API to query the Medidata system and build a report of everything we have received since the last query. Basically, we've written internal software based on Medidata's API that allows us to easily generate reports of newly received imaging in the queue, so we can quickly pinpoint what to upload into our system. Troy, is there anything you want to add on that?
Those are some great points and some powerful numbers. I think as systems become, touching on a point earlier, more and more integrated, you're going to see this time continuously speed up, and talk about a game changer in the industry. For example, the Medidata imaging system has direct PACS connections, so into the hospital imaging network, with over 600 institutions. This allows sites to easily find a patient in the hospital system, basically right-click on it, and send it into the IntelliMage system. We can then automatically route it and download it at Medical Metrics and make it available for reviewer. So traditionally what could take a few days can now take less than 5 minutes to get it from the hospital to in front of a radiologist's eyes. Now imagine they do a quick assessment, and if there's critical time needs, they can actually get that analysis back to the site and picture a scenario where that subject is still on site or still possibly with a tech near the scanner. They can actually rescan if they need or change that patient's plan of care based on that. So I think with things trending that way, we're in a great position to facilitate that workflow, which is a dream scenario for some studies.
Yeah, absolutely, Troy. I mean, the technology has really allowed us to move much more quickly in processing imaging. And this really brings us to the last point that I'd like to make. And this is an example where adopting the Medidata platform has allowed us to expand our services for quick turnaround and rapid image review. This example is for patient eligibility screening. In many cases, it's common to have imaging-based screening criteria that patients must meet in order to be enrolled in a study. The turnaround time for eligibility screening must be fast because sites can't wait long to receive feedback from the core lab on whether or not that patient is eligible for enrollment. For most studies, MMI will view images for eligibility screening the same day or the next day. This requirement for fast turnaround means that delays during the image transfer process must be minimized, and Medidata helps to address that issue. As a result of adopting the platform, we are currently meeting a 99% on-time delivery rate with the aid of the technology, and it's this kind of upgrade in technology that's really allowed us to expand our services and deliver higher value to our clients. So in summary, I can comment that the Medidata medical imaging platform has allowed us to deliver increased quality, value, security, and confidence to our clients. We have been able to assure complete protection of subject privacy, reduce our rate of discrepancies through automatic error detection methods and improve workflows, increase our throughput with improved efficiencies, and lastly, provide sponsors with improved access to their images and data. And the overall feedback that we're getting from our sponsors, from our CRO partners and others is that there's a significant value added from this technology integration. So I hope you found this information from the Core Lab's perspective to be useful. I'd be happy to answer any questions you may have, and at this point I'll hand things back to Carly to moderate any questions. Thank you so much.
Thanks so much. Now we'll move on to the Q&A. There's still time to submit your questions using the Q&A button in the lower left corner of your screen. We have lots of great questions already, and we'll try to get to as many as possible. So for our first question, you mentioned that you use Medidata to support non-clinical trial work. Can you comment further on that?
Yeah. So this is Nick. While MMI uses its own proprietary platform for managing clinical trial data, we do use the Medidata medical imaging platform to receive images and evaluate them and capture the results of our evaluations by physicians for non-clinical trial work, for product surveillance assessments. We've customized the platform so that depending on the type of product we are asked to evaluate, we can present a unique list of assessments to our radiologist, whether that's a total knee replacement or a total hip replacement, a shoulder, an elbow. We can customize the assessments given the particular type of product we're evaluating. And the radiologist can input a variety of information into predefined fields associated with that product. Additionally, the physician can add narrative commentary on the factors that may have contributed to the particular issue, and we report that information to the sponsor. This information can be held indefinitely in the system to support long-term data retention and future queries. And I would say this could be especially useful for product recalls or other kinds of projects where there is a need to collect and store, a wide variety of information about, the patient and the implant.
Great, thanks. And the next question on CRO's experience using the platform.
Yes, this is Troy. That's a great question. That seems to be one of our quickest growing segments. Keep in mind the Medidata imaging platform has a self-build feature set built in. So it's completely configurable. You can have a data management team at a CRO end to learn how to be enabled to build the simplest of studies, just get images from the sites to the labs, to even the more complex device ones we may see with different cohorts and screening committees and different adjudication portals. All that can be built and customized, including forms, your imaging visits, your requirements, all within the portal. So this seems like a natural extension for CROs to offer as another service outside of their traditional data management offerings. And for a lot of the smaller device companies, this is one less vendor they deal with, and things can be rolled up into more traditional services that a CRO offers. And I know Medical Metrics has a nice history as well with partnering with CROs.
Yeah, thanks, Troy. I mean, some CROs, that we work with have worked directly with the sponsor to obtain buy-in on the platform, so that when MMI has gotten engaged with the study, Medidata is already lined up and planned to be used, and this makes things much easier for us, so that the sponsor, the CRO, and the core lab are all on the same page about the use of the platform. I also know that some CROs and other service providers who help sponsors, for example, make site payments, they've used the system to verify that the imaging is in the system before approving payment to the site. So from my perspective, CROs have found the platform to be helpful for managing the study and for the CRO, the sponsor, the core lab to kind of work collaboratively on monitoring the flow of imaging.
Great, thanks. And our next question, what do you see most sponsors do with images at the end of studies?
Sure, we're big on providing options with metadata imaging. So we can do a few things. One is each core lab that's connected to our system, and there's 70 plus around the globe, has a piece of software called the director. This director checks in every 90 seconds and can download the images or send them to different destination or viewing tools. Well, that same concept can exist for the sponsor, and we have a lot of sponsors on the device community taking advantage of this. So imagine a piece of software that downloads all the images, form data, audit log in real time and stores it on local servers or other third-party servers that the sponsor can access at any time. We can also provide an official trial closeout where we'll collect all the data and electronically send it back to the sponsor with a certificate that the trial has been closed out. But I think, in keeping the theme of this session on technology, where we're seeing the biggest advancements is let's not just store the data, which metadata imaging can do through our data centers around the globe. Let's actually do something intelligent with them. Let's do something to help the greater good, the bigger company, other departments. So we have the concept of an image library. So the images, instead of just being stored, let's actually pull off all the DICOM headers, all the form data, completely de-identify the images, and store them in a library that's searchable, taggable, indexable, that anybody in your team or within your company can come into this library and search and say, show me a 70-year-old male from the EU with these conditions. And we'd bring up all the forms, de-identified all the images. This is a huge advantage for maybe your research team, your field team, for marketing, for training, to have this library of images that actually help tell a story and can help with other projects instead of them just being siloed or stored in a data center or in a cardboard box under somebody's desk.
Great, and this will be our final question. We had a lot of great questions today and couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. Is Medical Metrics working on other development and integration initiatives with Medidata?
Yeah, this is Nick. There are 2 or 3 main areas of development that we're working on and planning for. One is to expand our use of Medidata's API. This will further allow us to improve efficiencies when detecting and processing the new arrival of images. Over time, we believe we can further improve our turnaround times through tighter integration and communication with the Medidata platform through their API interface, so that we can query the system on the availability of images as they're flowing through the platform. Another area that we're working on is to implement more advanced inspections of the DICOM image headers. Many of the studies we are involved in require advanced MRI sequences. So to take one example, we're currently supporting all of the metal-on-metal 522 studies in the U.S. Metallic implants, create a lot of streak artifact, and so you have to develop specialized sequences to minimize that artifact. And to verify that sites are properly implementing these advanced sequences, we want specialized inspections to verify or assure compliance that the image acquisition protocol is being followed. Finally, a long-term objective is to make it possible to write the results of our analysis directly into the Medidata platform Again, tightening that integration and writing directly into the RAVE system so that we have good interoperability between our systems, between the core lab and EDC.
Great, thanks so much. Thanks to everyone for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speakers for participating and Medidata for presenting today's webinar. This webinar has been recorded. You'll be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events.